You know that feeling, right? When you bite into a slice of melon and it's just perfect? Sweet, juicy, everything you hope for? Well, what if I told you the secret to that perfection isn't just about sunshine and water? Nope. It's actually a super complex genetic story, a code hidden deep inside the plant's DNA. And today, we're going to be scientific detectives. We're going to crack that code. So what does a perfect melon have in common with a police investigation? I know, it sounds a little out there, but trust me, figuring out what makes a melon great is exactly like solving a crime. You've got to follow the clues, line up the suspects, and build a solid case to find out who's really running the show. And in our story, the crime scene, it's the melon plant itself.
Okay, so the big secret, the first major clue, is all about the plant's architecture. I mean, its physical shape. A melon's quality, we're talking sweetness, size, the whole deal, it all comes down to how the plant is built. And the key to that is something called lateral branches. You know, the little side shoots? That's where the fruit-bearing flowers actually grow. So it's pretty simple. More branches means the plant can catch more light, get more energy, and ultimately give you a bigger harvest of amazing melons. But what's telling those branches to grow in the first place? That's the real mystery we need to solve.
All right, Section 1, the genetic suspects. We're identifying a family of genes known for shaping plants. To figure out who's controlling the branching, scientists basically had to bring in a lineup of the usual suspects, a family of genes that has a reputation, a known track record for shaping how plants grow.
So, first things first, you gotta understand that not all genes are doing the same job. Some are like the workers on an assembly line, but others are the managers. These are called transcription factors. Just think of them as these tiny little supervisors inside every cell. Their entire job is to tell other genes when to turn on and when to turn off. They're the ones pulling all the strings from behind the scenes.
And that leads us right to our prime suspect family, the TCP genes. Now, this specific group of transcription factors has, let's say, a long rap sheet across the entire plant kingdom. They've been caught red-handed controlling everything from the shape of leaves to, and this is the big one for our case, lateral branching. The scientific detectives knew exactly where they needed to start looking.
Okay, on to our next section, inside the digital lab. This is all about how scientists track down the genetic culprits. Once they had their target group locked in, the scientists got right to work. But this wasn't your typical lab with bubbling beakers and test tubes. No, their entire investigation took place inside super powerful computers, where they sifted through just massive amounts of genetic data. And their method was basically pure digital forensics. Step 1 was a huge search. They scoured 5 major plant databases, looking for any sign of these TCP genes in melons. Then, Step 2, they had to confirm the identity of every single one by looking for a unique protein signature. It's like a genetic fingerprint. After that, Step 3, they profiled each suspect, analyzing all their characteristics. And finally, Step 4, they ran a background check, comparing these melon genes to their known relatives or accomplices in other plant species.
All right, new section, a lineup of 29 genes. Here, we're profiling the melon's TCP gene family. After all that digital digging, the investigators finally had their lineup. They had successfully identified every single member of the TCP family that was hiding out in the melon's genome.
So what was the final count?
29. That's right, 29 individual TCP genes. Every single one was a potential suspect in controlling how a melon plant grows. Now, with a lineup that big, the real detective work, the profiling, was really just getting started.
And here's the official rap sheet on our suspects. These 29 genes were scattered across 9 of the melon's 12 chromosomes, and they were mostly hanging out near the ends. And get this, 28 out of the 29 are predicted to work from inside the cell's nucleus. Now that is a critical clue. It means they're right there in the command center, in the perfect position to be calling all the shots.
Look, 29 suspects is still a lot to deal with. So the scientists did the smart thing and divided them into 2 groups based on their structure. You can think of them like 2 different gangs, each with its own specialty. You had Class 1 with 13 genes. They were mostly into general stuff like leaf growth and flower development. But then you had Class 2 with 16 genes, and their MO, they were known for regulating flowering and, you guessed it, lateral branching. All of a sudden, the case got a whole lot clearer. The investigators had their list of prime suspects.
So now we're cracking the branching code, following the trail to other species. The case was definitely getting warmer. To go from 16 suspects down to the real culprits, the scientists needed more direct evidence.
And the key, it turns out, wasn't in the melon itself, but in its relatives.
You know, this is actually a classic move in genetic investigations. If you want to figure out what a mysterious gene does in one plant, you find its counterpart in a relative that's already been studied to death. It's kind of like finding a reliable witness who's seen your suspect in action before, just in a different town.
So to find these connections, the team dug into shared genetic history. And as you can see from this chart, they found the strongest links to melon's closest relatives. Look at that, 49 connections to cucumber, 43 to tomato, but only 11 to a distant cousin like rice. The evidence was pointing in one clear direction. The melon's own family held the key.
And that was the breakthrough moment. Melons and cucumbers are evolutionary cousins. They're so closely related that a gene doing a specific job in a cucumber is very, very likely to be doing the exact same job in a melon. The cucumber genome was basically the Rosetta Stone they needed to crack the melon mystery.
So, this brought the whole entire investigation down to one single critical question. With all the clues gathered, all the evidence compiled, which of the melon's TCP genes were the real masterminds behind lateral branching?
And here they are, the prime suspects revealed. The investigation zeroed in on two main culprits, CmTCP03 and CmTCP27. And the evidence was just overwhelming. Why? Because these two melon genes were the direct one-to-one equivalents of genes that had already been proven to control branching in both cucumber and another really well-studied plant. It was a perfect match. Case closed.
Okay, on to our final section, the future on the vine. What does this genetic discovery actually mean for our food? So, the detectives found their genes, case closed. Awesome. But what does that mean for you and me? Why does solving this little genetic mystery even matter? Well, this is where all that science in the lab finally hits our dinner plates.
It's because once you know exactly which genes are pulling the levers, well, you can start to influence how they're pulled. This discovery isn't just some cool fact for a science textbook, it's a real, practical tool that breeders and farmers can use to literally create better melons.
And here's the real payoff, the why this matters part. By being able to select for plants that have the ideal versions of these genes, breeders can design melon plants with a better structure, and that leads directly to higher quality, sweeter fruit, and more of it. For farmers, that means bigger, more reliable harvests and lower costs. And for us, it means getting a more perfect melon every single time we buy one.
And this all leads to a really fascinating final thought, a question to chew on. We've cracked the code for branching. But as scientists start to map out the genes for sweetness, for disease resistance, for texture, are we heading towards a future where the next great melon isn't just discovered by chance in a field, but is actually blueprinted, gene by gene, in a lab first? It's definitely something to think about the next time you're enjoying that perfect slice.